All right. So today we are going to see about the die swap design versus the reference design in microarray technique. Now, one issue for two color array, that means when you are using psi3 or psi5 die in a particular experiment, so then uh, die bias effects are introduced. Okay, uh, because of different chemistry of these two die. It is important to control for this die bias in the design of your experiment, and that's why you can use uh, die swap design or a reference design. Now in this diagram you can see that in a die swap design, the same pairs of samples, that is test and control. So these are pair samples, right? Control sample versus test sample are compared twice with the die assignment reversed in the second hybridization. So if you are going to use one color for control in one experiment, so in second experiment you need to do uh, the you need to use that same color for the test sample. So you are just swapping the die color. Okay, so you need to perform the experiment twice. Okay, but this is costly. You can understand that you need to use two chips for one particular test. So maximum time, the reference design is followed, where each experimental sample is hybridized against a common reference sample. Okay, now for that you don't need to use two uh, what is it, two chips, but you can make your test sample and control sample with the same die, and you use another set of sample as a reference so that you can compare these two with this sample. Okay, so this kind of techniques are commonly used.
Sir,
Now
why we need to do this? I mean, this die swap design.
to to overcome the problem of die biases, because you are using two die, so the fluorescence intensity which are coming out from that samples, that will be different. Okay, so to minimize the error, you need to check whether you are getting such uh, values or such fluorescence intensity for this die, or it is for the expression value or the gene expression itself. To cross check that, you are swapping the die into sample, one control sample and test sample. Okay,
Yes, sir.
all right. So next is okay. I will come to this later, because after performing the microarray data analysis, so you will present that data in a format of matrix again. So where the columns are correspond to some conditions. Okay, so C1, C2, C3, C4, these are the conditions, and these are the genes, gene A, B, C, D, E. So the row wise, if you can see that row wise, these are the expression values for that particular gene in a condition. Okay, so we will present that thing in this format, and this is known as gene expression profile. Gene expression profile. We will come to this later also. But before that, there are some tools available for performing different uh, components of data analysis. So these are very widely used tools. So I have included here. So quality assessment you can do using box plot or MA plot. Normalization, it can be done by many algorithms, RMA, GCRMA, MAS5. Differential analysis can be done by Lima or AFI package. which are available in bioconductor, multiple testing, Benjamin Hedgehog or FDR analysis. Clustering can be done by hierarchical or k-means algorithm. Visualization can be done by heat map or histogram, and function analysis of the genes can be done by KEGG or geo analysis or GACA analysis. Okay, so these are the commonly used microarray data analysis tool. And now you can ask me that then you are saying that different normalization techniques are available. So which one need to be followed? So that is the question. If you are asking me, I don't have any specific answer, but it is suggested that you need to cross check with your result or with your data by different normalization techniques. Okay, if you find that okay, it is your gene sets are overlapped by using two kind of techniques, so then you can use any one. Otherwise, what you can do, you can combine these two types of normalization techniques, and then you can stringent your data sets. Okay, so that is the protocol. You need to play around with your data to get maximum important genes from the data set. Now, the first step is data normalization. If you have seen that data normalization is required. Yes. Where is that? Yes. So after quality control, normalization is required. Now the question is, what is normalization? And how we can perform that normalization steps. Okay.
So normalization is to reduce the size of the data so that we can fit in between two axes, and this can be done by square root transformation or log normal transformation, like that kind of stuff.
So are we going to reduce the data sets? So then what is the use of
No. Not reducing the data set. We are just reducing the values.
uh, right? So we are
We are not
scaling.
we are not reducing the dimension. Yes, we're not reducing the dimension. We are just
Right.
reducing the scale so that it can be fit in two axes very easily. So that
Right.
it is easily transformed. I mean, easily visualized.
Right. Suppose one experiment, you are getting this kind of raw data, which is exponential um curve you are getting, but you it is not very much easily quantifiable. Okay, so for that you can do log transform technique, and then the data will be spread out like this, and then we can see that whether the data is good or bad. That kind of uh, comparison we can do after log transform. Okay, so these are the technique. I am not going to discuss this. uh, we will come to this later also. uh, uh, okay, here you will find that the same terminologies are used for scaling or calibration or normalization. So both all these terms are equally applied, and it describes the process of removing or minimizing the non-biological variations in measured signal intensity levels so that biological differences in gene expression can be appropriately detected. Okay. Now, next question is, then what problems we will face? How do we detect the biases? And which genes should we use for estimating the biases among the chips or channels? So these are the questions arise during the normalization stage. And how do we remove these biases? So for that, we can use different types of genes in the genes chipset, right? So we can include housekeeping genes. Housekeeping genes. Housekeeping genes are what? You have heard about this housekeeping genes. Housekeeping genes are such genes which which are expressed in
which
all the
control the
conditions.
other cell.
No, that expressed equally expressed in every samples in every cell. Okay, suppose some transcription factor gene, so that is required in every cell. So those are treated as housekeeping genes. Now, if you are incorporating this kind of housekeeping genes in your chip, so in latter case during the analysis time, you can compare that whether your uh, other genes are expressed more or less than those housekeeping genes. Okay, so this kind of housekeeping genes or some spike in controls. So those spike in controls are from other organisms. Suppose you are making some human chip, so then you can include some E. coli gene sets also in the chip to check that whether there is any uh, what I say, any changes in that spike in control. So whether the cross hybridization is performing in those regions. So that kind of biases can be detected using such techniques. Okay. And there are multiple. Okay, so I missed one point. So when I am talking about that it is reducing the non-biological variations, so what are the sources of such non-biological variations? So you should keep in mind that dye bias. So I told you that due to the different nature of the chemicals which we are using as different dyes, Psi3, Psi5, so they have different heat and light sensitivity, uh, so that can generate some biasness. Then amount of labeled cDNA that is hybridized to each channel, okay, that can influence. But sometimes some cDNA will vary, the amount of cDNA which are produced from the mRNA level, so that can be varied, and due to that there will be some variation. And another variations like the hybridization condition, so that can be varied. Okay, scanning condition can be changed. So these are the non-biological variations. So we can minimize this thing by normalization technique. Okay. Now there are multiple normalization methods. One is global normalization method and another is quantile normalization method. Most of the time we use this kind of quantile normalization. You know, these are known as box plot. So if you are if you have started your data analysis first time, so you will find that such kind of box plots are generated. Then after applying the quantile normalization, you will see that all the arrays are arranged in this fashion. Okay, now see, this is known as box plot, and each of these box have some bar at the mid side, so that is known as the median value. Okay, now we cannot compare this boxes which are actually representing each of the data sets by this way, right? All are not equivalent position. We cannot infer any conclusion from such type of arrangement. But if we arrange the boxes in this pattern, so then we can check that whether which data sets is vary mostly, or which data set we can include or which data set we can exclude from the experiment analysis further. Okay, so normal quantile normalization technique, after quantile normalization technique you will get this kind of arrangement of data sets. Another is intensity dependent normalization. So So I am not going to discuss about the algorithms here, but you should remember the techniques or some of the algorithm name. Say for example, one algorithm is RMA, another is MAS5, another is LOES, okay, or GCRMA. So, so any one of the normalization technique you should follow for data analysis purpose. Okay, next, after normalization technique, we should identify the differentially expressed genes. So that is our main aim. That when we are designing some experiment, suppose one treatment group and one control group we are uh, comparing. So our aim is to identify the genes that are differentially expressed in those two conditions, right? And for that, we need to consider the number of replicates also. Now, when we are talking about number of replicates, so in any kind of experimental designing, you should know the details of the number of replicates, right? How you can choose that how many replicates do you need. So what do you think when you are going to perform any kind of experimental techniques, like your in your experiment lab, biochemistry lab, when you are performing any kind of DNS test or Bradford assay, so you are going to use some replicates, isn't it? Yes or no?
Yes, and we are
Yes, sir.
usually do it in we usually replicate in 3 times.
Right, right. Now the question is, what type of replicates are those? Because in any kind of experimental designing, there are two replicates actually used. One is known as technical replicates and another is known as biological replicates. Okay, so what is the difference between these two? That you should know. What is the difference between the technical replicates and biological replicates? Do you have any idea about this two? Okay, do you have, do you know the difference between biological and technical replicates? What are these?
So technical replicates means we need to repeat or replicate the entire technical process. But biological replicate means we need to perform it on more similar kind of individuals.
Okay, but why?
To make it more error free, to reduce the standard error.
Uh, then why two types of replicates are required?
Because there can be some sort of technical issues, like if we are performing some experiment with the help of some machine, every machine have some sort of error index. So that's why, to reduce that error index, we need to perform technical uh, replications. And
Right,
also there are different kinds of uh, what to say, like if we work on bacteria, there are different kinds of strains. And if you work on some other organisms, not every organisms are exactly alike.
right,
That's why also.
right. Good. So let's see this. What is
Sir, please
explaining?
explain this thing. I can't understand anything.
Right. So, so this is the way how we can produce the biological replicates and technical replicates. Now, suppose you are interested to study from leaves of one plant. Okay, so if you can collect the initial sample, so that is A and B, these two leaves you have collected. Now these are these will be treated as biological replicates because those are taken from the same plant. Okay. Now, when you are going to measure something, suppose any biochemical component or chlorophyll from that leaf you are trying to measure. So, if you are again dividing that sample into two appendrops, so those will be treated as your technical replicates. Okay.
Dividing from the same biological sample
Right. So from
without
here,
any changes. I mean, without any chemical treatment or anything.
no, without any chemical treatment or anything, you have just divided your sample. Okay. So that will be treated as your technical replicate.
Okay, sir.
Now, biological replicates are used to sample of individuals from a population in order to make inferences about the population. Okay. So what variations across the population, that kind of information you can get from the biological replicate. And technical replicates address the measurement, the error of the assay. As he explained that every instrument or every step, suppose you are performing 10 steps in an experiment. So every time you there might be some pipetting error if you are using any pipette manually, right? So technical replicates, if you are performing technical replicates and then you do the same experiment, so after that if you are taking the average of that thing, so you are minimizing the error. Okay. Measurement error you are minimizing by technical replicate. Now, why this kind of uh, experiments or replicates are useful? So you can see that in this slide, in different omics study, we are going to use number of samples, number of replicates, right? So minimum number, as he said, that we took 3 samples at least for better result, better data. So here 2 examples have been given for treatment with A. So these will be treated as biological replicates, these 2 mice, and the mice which are treated with B sample, so those are treated as biological replicates for the second set of mice. Okay. Now from each mice you can take blood, in you can even mark them as technical replicates. Okay. And then you can perform different combinations for performing microarray. Like you can take this sample from A treated mice and you can take another sample from B treated mice. Okay, you can perform one comparison with this A and B array sample. Okay, so
the samples are same. I mean, the red and the green one samples are same.
oh no, that technical samples you can use for different uh, colors, means psi 3 or psi 5 if you are going to use microarray. So you can tag those sample by different uh, dye. Okay, different color dye. That's why it is shown in red and in green. But these
Sir,
are,
I mean, these 2 samples are blood here.
yes, taken from blood, but those, not blood means what? Blood means if you are going to perform microarray, so you need to isolate the RNA, you need to uh, make it cDNA, and you need to add the dye psi 3 or psi 5, then only you can perform the hybridization technique, right? So even if you are going to take collect the blood first, so you need to do that steps also. Okay. Is it clear what I am trying to say? That here the blood sample is collected, but the 2 samples will be treated as technical replicates when you are uh, taking that sample from the same biological replicate. All
Okay,
right. Okay. Now we are going back to that slide. What is that? Yes. So next we will see about the fold change. Now suppose we are using this 3 sets of control and 3 sets of treatment group. Okay. Now for a particular gene, we are getting some value like 90, 100, 110. And for the second group, that is treated group, 190, 200, 210, like this. Now we need to compare that whether the gene is expressed more in treated group or not, right? So that is our aim. We need to conclude that whether a gene is expressed more or less in compared to the control sample. So for that, we need to measure that thing. There are some ways to measure that thing. One simple way is fold change. Now, what is fold change? Have you heard about this term, fold change, earlier? Probably you have done. Some
no,
of you
sir.
have done artificial, right? So during that also, during that technique also, you have calculated the fold change. Okay. So in simple way, fold change means the ratio. Fold change is the measure that describe that how much a quantity changes between an original and a subsequent measurement. Suppose you are trying to find out, you are trying to compare this control versus treatment group, right? So then you need to find out the ratio between these two quantities. Okay. So 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 fold change will be calculated by a ratio. Suppose treatment divided by control. This value will be the fold change. Treatment divided by control. That is the easiest way. Suppose I am giving you one task that suppose after this treatment, okay, so the gene expression value change from 30 to 60. Okay, suppose earlier the control, the gene was expressed, or simple take this example. So in control, on an average, the gene 1 expression value shows that 100 mean expression value. Now after treatment, that gene shows 200 unit. So then what will be the fold change for that gene?
Sir, I can't understand. Please repeat.
The question is, when you are trying to figure out that how this gene 1, can you see this slide, fold change?
Yes, sir. Yes, sir.
So my question is, suppose this gene 1 is expressed in the control set, this 3 condition, and this treatment set in this 3 samples, T1, T2, T3. Okay. Now these are the values given: 90, 100, 110, 190, 200, 210. Now you have calculated the mean value for those expressions, that is 100 for the control sets and 200 for the treatment set for gene 1. Okay. Now, if I ask you that, now tell me what will be the fold change in compared to control in treatment group. So how you can calculate that thing? Very basic mathematics.
The expression is become doubled. Here is 100 and here is 200.
Yes. So how you can measure that thing? How you will calculate that thing? So you are dividing this 200 by 100, isn't it?
Yes, sir. Yes, sir.
So that is a ratio. 200 divided by 100. That is a ratio, and this is known as fold change. Okay. So for n number of genes, you can calculate the fold change for comparison purpose. But
Sir, how can I measure the expressions of these values?
you mean these values?
Yes, sir. Yes, sir.
So those are coming from the data. Data means the raw data. That is from the signal intensity from the microarray chip.
Okay, okay.
Okay. That cell.cl file, from that .cl file, you are extracting that pixel values, and those are the signal intensity values. Okay. Another technique is t-test. So you can see that if you use this formula, this is a statistical test. So then you will find that for each gene you will have some t score. Okay, and from that t score you can check that whether the gene is overexpressed or not. That kind of conclusion you can do. Okay, so that is another technique. The easiest way is fold change. Another way is to perform t-test. Okay, then obviously there will be some statistical test to be performed for getting the um, for getting the more accurate value. So you know that statistical tests are performed on this test is, and you can get the p value also, and based on that p value you can determine whether your test is good or bad. Okay, so I'm not going to discuss about the multiple testing, but multiple testing need to be done when you are using multiple samples, right? So there are certain corrections for uh, multiple testing. Those are Bonferroni or Benjamini-Hedgehoff method. Multiple tools are available to calculate, to find out the differential genes. One is SAM tools. Okay, significance analysis of microarray. This is a diagram where you can see that using this microarray technique, we can also find out the different isoforms, okay, which are not very easy to find out, but designing with some complicated experiment, this microarray techniques can also detect some isoforms also. So if you are interested, you can look for that, how the microarrays can be used in isoform discovery. Okay, so today's class time is over. So next day we will discuss about some practical aspects of microarray data analysis, how we can perform microarray data analysis from GEO site itself, GEO, gene expression omnibus, with the help of GEO2R2. Okay, and if you have any doubt, you can ask me uh, the next class also. So tomorrow we will see again.